Assalamu alaikum. Let me welcome you to a presentation on meninges, the covering of the brain on behalf of Ahsan Academy. In this presentation, I will inshallah uh, cover the concept of covering of central nervous system, then the components of those coverings and the foldings made by those coverings and spaces between those coverings with the clinical correlation at every stages. There are three protective membranes surrounding the brain and spinal cord. They are the dura mater, the arachnoid mater, and the pia mater. Of these last two, that is arachnoid matter and pia mater, together known as leptomeninges. Lepto means thin. So these are the thin membranes, and the first one is very thick. That's why it is known as pachythin. Pachy means thick. Let us start with the dura mater. It is very thick, tough, and outer covering of the brain, and it has two layers. In case of the cranial dura mater, we will discuss the spinal one later subsequently. So the cranial dura mater means it is present within the cranial cavity and covers the brain, and it is very thick, tough, and outer covering. It comprises of endosteal layer, which is outside, and meningeal layer, which is inside. And this meningeal layer is actually two meninges, and the endosteal layer actually covers the inner side of the cranial bones and fused with the endosteum, means the periosteum of the bone. This one by the arrow indicating the endosteal layer, and you can see the another layer that is indicated by the arrow, and it is the red line is indicating the direction, making the fold inside a line between the parts of the brain. This one in the figure is endosteal layer of the cranial dura mater, and this remain fused with the endosteum of the cranial bones. And the second one, this one is indicating the meningeal layer. So you can follow the meningeal layer. Actually, the venous dural venous sinuses lies between these two layers, endosteal and meningeal. So this is very closely related to the arachnoid mater below, and it makes the fold, as I have said earlier, and this meningeal layer continues as the spinal dura mater at the level of the foramen magnum. So beyond the foramen magnum, the endosteal layer doesn't continue, and it becomes continuous with the periosteum through the foramen magnum. The periosteum lies outside. Let us discuss the dural folds. These are actually going as the septum, but it is not complete. It's divide the cranial cavity into incomplete chambers. There are four folds, and we can classify them. Falx cerebri, falx cerebelli. These are vertical folds, and tentorium cerebelli, as well as the dos diaphragma sella. Those two are the transverse folds. You can see this is the falx cerebri. It is sickle shaped and going between the two layers of the cerebrum as shown in the figure. And I have drawn actually with dotted line because this remain under cover of this tentorium, goes below the falx cerebri, and these are relatively small, lying between the hemisphere of the cerebellum as indicated by the arrow. These transverse dural folds are known as tentorium cerebelli, and you see a small dural fold that covers the body of the sphenoid bone lies over the hypophyseal fossa is diaphragm sella. Falx cerebri in the left figure, the left lower figure, if you see, this is the falx cerebri, and it's a sickle shelled and vertically lies. It is anteriorly attached to the crista galli of the ethmoid bone. You can see this in the photograph, and just anterior to this, the continuation is the internal frontal crest present inside the frontal bone. So this anteriorly, the falx cerebri is attached to this, and posteriorly it continues, and superiorly it is remaining attached to the margin of the superior sagittal sulcus. Posteriorly, you can see this falx cerebri has a wide margin, and this margin fuses with the tentorium cerebelli at the midline. Within the falx cerebri, close to its margin, lies some venous sinuses. This is the area of the superior sagittal sinus that starts anteriorly and gradually become larger and wider and goes posteriorly. Similarly, the inferior sagittal sinus lies close to the lower border, and it starts anteriorly and gradually become wider posteriorly. The junction between the falx cerebri posteriorly with the tentorium cerebri, this junction contains a straight sinus running from anterior to posterior. Within the falx cerebri, you can see the small veins that runs and open into either superior and inferior sagittal sinus and connects them. Falx cerebelli, this is also median fold, lies between the hemisphere of the cerebellum. Let me show the fold by changing the color and indicating. This is the internal occipital protuberance, and then internal occipital crest running inferiorly. So this starts from the internal occipital protuberance and goes up to the foramen magnum. This falx cerebelli remain attached to this line. You can see this, the red shadow is indicating this one. So superiorly it fused to the posterior part of the tentorium cerebelli, and it is much smaller than the falx cerebri. It lies between the two hemispheres of the cerebellum. One is the tentorium cerebelli, you can see. So let us see the attachment of the tentorium cerebelli. This is the tentorium cerebelli. This is tent-like, means placed almost horizontally, and I'm showing by different photographs. The one this is labeled in the left upper figure. Now look at the figures below that. You can see the two red arrow indicating the tentorium cerebelli. This is running transversely and have a deficiency anteriorly. Let us see the attachment of this tentorium cerebelli. This is the sphenoidal body, and posteriorly this is having posterior clinoid process. That is, I am making a round red mark. This is the posterior clinoid process. This has two on either side. One is marked, the opposite side is also having. And let me show the margin, the red color indicating the margin anteriorly. This is single line. Actually this is attached to the margin of the petrosal sinus, but posteriorly the transverse sinus is much wider, so sulcus is much wider, so it has two leaves. So posteriorly the tentorium cerebelli splits to remain attached to the margins of the transverse sulcus. Now let me indicate the tentorium cerebelli by yellow color. This is the tentorium cerebelli proceeding from the back to anterior, but it is having a large gap, and this margin is known as tentorial notch. as indicated by the red dotted line. So the tentorial notch uh, allows the part of the brain to pass through this to reach the posterior cranial fossa. You can see here I'll change the color to show the tentorium. Yellow color shade is showing that this is the position of tentorium. So this is lying between the cerebrum and cerebellum. It separates the posterior part of the cerebrum from the upper surface of the cerebellum. Diaphragm ocella, this is another transverse fold but much smaller, and it lies over the sella turcica, means that is present in the superior surface of the body of the sphenoid bone. Let me indicate, this is hypophyseal fossa. This is the concave part of the sella turcica. It makes a convex anterior area and convex posterior area, so together known as sella turcica. So this makes a roof over the hypophyseal fossa and attached to the anterior and posterior part of the sella turcica. You can see this is the diaphragm ocella, and you can see a rounded area. This is an opening. Through this opening, the below this lies the pituitary gland. So pituitary gland is continuous with the hypothalamus by the infundibulum. So infundibulum passes through this opening. Let me show this. This is opening of the infundibulum. So diaphragm cella has one opening, that is the opening for the infundibulum, and tentorium cerebri has hiatus. Through this, the forebrain is connected to the hindbrain present in the posterior cranial fossa by the midbrain. Midbrain lies at the level of tentorium hiatus of the tentorium cerebelli. These folds divide the cranial cavity into incomplete chambers. So these walls are not complete. So these chambers are freely connected with each other. And whenever there is movement of the head, it restricts the movement of the brain, which is not solid. It is relatively solid and soft. So because of the inertia of the movement, certain movement of the head may twist the brain, and this folding prevents that. It carries the brain with the head, even the head is certainly moving. Let us discuss the artery supply of the dura mater. That is clinically very important. We will discuss one by one. This major artery is arising from the maxillary artery. This is not a middle meningeal artery. This one is maxillary artery, and this artery arises outside the cranial cavity in the infratemporal fossa. So this crosses a foramen spinosum to enter into the cranial cavity. This is foramen spinosum, and immediately it divides into anterior and posterior division. You can see the anterior division passes anteriorly, and that is clinically very important. This this area crosses the pterion area internally. So we will special emphasis to this pterion area in the next slide, inshallah. So anterior meningeal artery is a very small artery. This arising from the ethmoidal artery. It also supplied by the posterior meningeal artery. This may arise from different sources and maybe multiple. It arises from the ascending pharyngeal, from occipital, and from the vertebral artery. There may be multiple on one side. This pterion, let me show the area. This is the sphenoid bone, greater wing of the sphenoid bone. This one is the frontal bone. This is parietal bone, anterior angle is there, and this is squamous part of the temporal bone. Look at this, all these four bones meet at this H-shaped junction. So this is known as pterion, and look, this area is relatively thin and weak. Here, if we see internally, the anterior division of the middle meningeal artery lies very close to the sternum, even if this may be within the bony tunnel. So, any problem with the joint or thin bone here may rupture this artery. Like this is the middle meningeal artery, and you can see the how close this middle meningeal artery is in near the tibia. So any blow, like this is the ball hitting this area, so any blow may cause rupture of the anterior division of the middle meningeal artery because of the bony fracture or dislocation of the joint. So this rupture of the middle meningeal artery will cause severe bleeding just outside the dura mater. So this is known as AP. AP means over, means outside. Epidural hemorrhage. So this can cause severe problem, and even patient can die if not taken care of. This is the dura mater. If it is injured along with the injury of the blood vessels, then this blood will be in the subdural space, means between dura mater and arachnoid mater. That will also cause pressure effect on the brain and leading to severe problem including death. This is the subdural hematoma shown by the arrow. Second layer from outside is the arachnoid mater. You can see this, the dura mater is separated, and you can see the arachnoid mater, and through that arachnoid mater you can see the brain substance. So this is very delicate, but it is impermeable to fluid and electrolytes. This lies internal to the dura mater, and it is connected to the pia mater by several delicate strands of fibrous tissue. This is dense connective tissue bands. These are known as trabeculae. Plural number is trabeculae, singular number is trabeculum. I will go in detail of this connection. So this is the endosteal layer of the dura mater. This is meningeal layer of dura mater. So under that is the arachnoid mater. So this is arachnoid mater, and this one is the pia mater. You can see several connective tissue bands. These are the trabeculae connecting this arachnoid and pia mater. This is the photograph, relatively larger view. This is skull. This is the dura mater. You can see the arachnoid mater, and at the depth very close to the brain is the pia mater. So in between, this space is filled up with connecting bands. These connecting bands are enlarged here. These are the connecting bands composed of dense connective tissue. So you cannot separate arachnoid mater from the pia mater without splitting this trabeculum. If not, then pia mater will be separated, injuring the brain. Arachnoid villi are the special features specially made to send back the cerebrospinal fluid from the subarachnoid space to the venous circulation. These are the projection inside the venous sinuses. This is projection of the arachnoid mater. So mostly it is present along the superior sagittal sinus, and there may be cluster of or clamps of arachnoid villi. This clamp is known as arachnoid granulation, and this is so large that we can easily see them by naked eye. So this arachnoid granulation, you can see this is the arachnoid granulation having so many villi on its surface. So villi are the individual projection, and the cluster is or clamp is known as arachnoid granulation. You can see this is one villus and this is actually projection from the arachnoid matter and carry the space under the arachnoid matter, and this is responsible for return of the CSF fluid to the venous circulation. This is the dura mater and this is the arachnoid mater. So arachnoid mater piercing the dura mater and getting inside the sinuses. This is arachnoid villi. Villus is the singular number. Pyometer. This is very thin and vascular membrane. It is tightly adherent to the brain and it is not possible to dissect it out easily from the brain surface. This continue with the surface and remain congruent with the elevation and depressions of the cerebral surface and other brain areas. It goes with the lobes, fissures and so on. So it is covering the gyrus. You can see the elevation is called gyrus and the depression on the surface of the cerebrum is known as sulcus, and plural is. You can see this is the dura mater, this is the arachnoid mater, and these are the connective tissue trabeculum, and inside that this is the innermost layer is the pyometer that follows. It is closely adherent to the brain and follows the contour, means any elevation, any depression, any fissure followed by the pyometer. It also follows the blood vessel. This is artery and that one is the vein. Blue color is the vein to some extent, and this continuation membrane become discontinuous and ceases. So so it remains close to the artery or vein, close to the surface. The dura mater is supplied by the sensory nerve, but it is not supplying the pyometer and arachnoid mater. So nerve supply is restricted to the dura mater, and this sensory supply is also with the wall of the blood vessels in that area. The sensory supply is from the three divisions of the trigeminal nerve. Previously also I have discussed that trigeminal has three divisions supplying the face: ophthalmic division, maxillary division and mandibular division. So it extends up to the scalp region, a vault of the skull, covering the vault of the skull, and posteriorly two spinal nerves, cervical second and cervical third, supplies the skull. So deep to this, the dura mater is also supplied by the same nerves. You can see this figure. This is the demarcation point, and this is actually indicating the level of transverse sulcus. So outside on the back, you can you can feel the external occipital protuberance. This is the area superior to this area. You can see this one, this superior to this transverse sinus or sulcus, and below this tentorium, the area is supplied by, above is supplied by the trigeminal nerve, three divisions, and below it is supplied by the, you can see this is cranial nerves. So up to this ophthalmic division actually continues more, and mandibular division also is up to this area, and maxillary division in between proceeds up to this external occipital protuberance. So any problem here, let me discuss it detail. This is the skull bone, and these are the dura maters, two layers of the dura mater, and the space outside the dura mater is epidural space. This is the arachnoid mater. So let me shade the area with the yellow color. This area is the subdural space. So dura mater is considered at the middle. Outside this is the epidural space, and deep to this is the subdural space. Most important space is between arachnoid matter and the pia matter. Let me show this one. This area is a subarachnoid space. It is deep to the arachnoid matter. So this space is traversed by many connective tissue bands. Those are trabeculae, and this connects the arachnoid matter with the pia matter crossing this space. The above two means epidural space and subdural space are potential space. So you can split them, but naturally there is no gap. But we can separate at these layers at this plane. But the real space exists between the arachnoid matter and the pia matter, which is known as subarachnoid space. That space is important for anatomical purpose as well as clinical purpose. So let us discuss the subarachnoid space. This contains the blood vessels and the nerves. In this space, it contains the fluid. You can see I have shaded with yellow color. You can see this is the area. I have filled it with the color, and this space is filled up with trabeculae running from the arachnoid to pia mater. You can see it in this left figure clearly. So these are the contents. So a fluid media, CSF, cerebrospinal fluid, is very important consideration anatomically, functionally, as well as clinically. Subarachnoid cisterns, in some places the subarachnoid spaces are widened. These dilated areas are known as cistern. Cistern is the singular number, AE if you add is the plural number. So what are those? These are the interpeduncular cistern. The arrow is indicating this is the area interpeduncular cistern. Second is cisterna ponti, just at the junction of the pons and medulla oblongata anteriorly. This space is situated, and on the posterior aspect, cerebellomedullary cistern is there. This cistern is known as cisterna magna, and this one is clinically most important area. So to collect the CSF, we actually collect the CSF to do some chemical analysis to identify certain diseases or conditions. So you can see this arrow is indicating the area, and it is approachable from the back through the foramen magnum. So you can easily approach with the needle through the posterior part, and that needle should pass through the foramen magnum to enter into the space, and from here we can easily collect the cerebrospinal fluid, and this procedure usually done in case of babies. Bleeding may occur at different pains. So let us discuss the different types of bleeding and its causes. So this one is the first form. I can show you. This is the yellow layer is indicating the dura mater. You have seen close to the, in case of the terion injury, deep to this lies the anterior division of the meningeal artery. You see the white arrow is showing the arterial rupture. So if it ruptures in the epidural space, there will be epidural hemorrhage, and that will split the dura mater from the bone and will cause a pressure over the bone, as you have seen here, causing a problem of that size, and ultimately because of the raised intracranial pressure the person may die. The second bleeding, the white arrow is showing the venous rupture. You see the arachnoid matter is very vascular. The blood vessels lies even between the arachnoid matter and the dura mater. This plane is an area where blood pressure veins may be ruptured. So the blood will be collected between, you see this marking is the dura mater, the yellow line, and you can see the subarachnoid space. A network is shown, and just outer layer is the arachnoid matter. So blood has, this is venous blood. A blue color is indicating the venous blood is splitting the dura mater and arachnoid matter and accumulated in this area. So this is known as subdural hemorrhage. Let us see this one. This is the subarachnoid space indicated by the network of trabeculum. So bleeding has occurred, and this is spreading through a fluid media CSF. So this type of hemorrhage is known as subarachnoid hemorrhage. So between layers in the spaces, three types of hemorrhage are possible: epidural, subdural, and subarachnoid. If it occurs within the brain substance, this is known as intracerebral hemorrhage. So we have discussed four types of hemorrhages: epidural, outside dura mater; subdural, between dura mater and arachnoid mater; between arachnoid mater and the pia mater. This is a subarachnoid space. If it occurs in the subarachnoid space, it is called subarachnoid hemorrhage. So epidural hemorrhage and subdural hemorrhage, it remains localized and causes pressure over the bone. And in case of subarachnoid hemorrhage, as this contains CSF, so blood spreads throughout the surface and it carried by the CSF everywhere. In case of intracerebral hemorrhage, usually it occurs in a limited place, but it diffuses and spreads as the blood volume increases. It spreads to the different parts of the brain. You can see the temporal region, and close to the pterion, around the pterion area, there is swelling. So it indicates that this hemorrhage may be in the scalp region, or this may be fracture of the pterion leading to bleeding, and the bleeding has come out through the fracture area. So cranial dura we have discussed. Now the cranial meninges actually we have discussed. Let us discuss the meninges that covers the spinal cord. This is known as spinal meninges. So all the three layers are there: dura mater, arachnoid mater, and pia mater. What is the difference? You see, the spinal dura is different. Spinal dura comprises of the single layer, and it is continuation of the meningeal layer of the cranial dura. At the level of the foramen magnum, the indicated by the dot line and the arrow indicated below, the meningeal layer continues and become the spinal dura that goes up to the second sacral vertebra, all the three layers, but pia mater continues more. So spinal cord is here present up to the lumbar, initial lumbar level, lumbar 1, or in children it may be up to lumbar 3, but the meninges continues further. You can see this is the sacral canal, and a conical shape extension has gone beyond the level of the lumbar, enter into the sacral canal up to the second sacral vertebral body. Arachnoid mater and paramater are a continuation respective layers. Means arachnoid mater goes to the spinal cord as arachnoid mater, and pia mater the same way. The pia mater of the brain is closely attached to the brain, so pia mater of the spinal cord remains very closely or intimately related to the spinal cord substance. This is the arachnoid mater, and this is the pia mater. The layer reflected outside is the dura mater, which is visible in the upper part. How does it terminate? This one is labeled as lumbar 1 vertebra, and at the level of sacral 2 is shown by S2 labeling. You can see this is the lumbar 1 level, and this termination of the spinal cord, a conical termination, is known as conus medullaris. So conus medullaris is tightly adherent by the pyometer. So pyometer at the tip become thin and continue downward. This is known as phylum terminale. So pyometer beyond the spinal cord continues as a thread-like structure and continues further till the coccyx. You can see this root of the spinal nerve at every level. So beyond that level, the roots forms the chordae equina. So chordae equina lies outside the pyometer and inside the arachnoid matter in the subarachnoid space. If you take section, you can see the coverings are the dura mater and arachnoid matter. Inside there are so many root layers and roots. This is chordae equina, and inside one greenish structure is there. This is phylum terminale, means a thin thread formed by the pyometer. So inside all the roots are within the cerebrospinal fluid, because this space is subarachnoid space. So beyond the lumbar 1, the subarachnoid space continues, which is very wide and contains the spinal roots, nerve roots. This is the coccyx posterior surface. So phylum terminalis, that is the continuation of the parameter, is fused here, but the dura mater and arachnoid matter ends at the level of the sacral 2. So subarachnoid space continues up to that level, and beyond that level only the phylum terminalis, that is the continuation of the parameter, is there. So let me repeat it. At the lumbar 1, the spinal cord ends in the adult, then the nerve root continues and hanging from that up to the end of the sacral canal, and these roots together known as chordae equina. So this chordae equina lies within the cerebrospinal fluid. So outside that, the dura mater and pyometer remains closer to the bone. So the subarachnoid space become very wider here in this place, and if we place needle below the lumbar 2, there is no chance of injuring the spinal cord. So we can collect fluid from this subarachnoid space. Above this level, you see spinal cord is there and occupying almost total area and very close to the arachnoid matter. So if we place the needle, there is every chance to injure the spinal cord. This is dura mater and arachnoid matter shown inside. So inside that is the pyometer. So pyometer on either side makes a conical projection. This is known as ligamentum denticulatum. This is present at every level between the roots of the spinal nerve. So it extends up to the level of the existence of a spinal cord. So this ends beyond the lumbar 1. So up to lumbar 1, we will find that the pyometer from the lateral surface conical projection is there that penetrates the arachnoid matter and fused with the dura mater. So this actually keeping the spinal cord in the middle. So this is helping to suspend the spinal cord in the middle, because you see dura mater is very closely related to the bone, and it is almost fused to the bone, and this denticulatum, ligamentum denticulatum, is attached to the dura mater and is penetrating the pyometer, fused with the lateral side of the spinal cord at every level. So this is keeping the spinal cord suspended in the spinal canal. The dura mater is stained here green, and pink colored one is the arachnoid matter. Both this layer continues with the spinal nerve up to the intervertebral foramen. So the spinal nerve carries this sheath up to the level of the intervertebral foramen. So beyond that, this spinal nerve is covered by the epineurium, means the outer connective tissue sheath. So these two dura mater and arachnoid mater fused with the outer connective tissue sheath of the nerve. The coverings of the central nervous system provides protection because it's a tough covering sheath all over the brain and spinal cord. It divides the cranial cavity into compartments, which helps this to protect the brain during the movement of the head. The brain substance is not solid, it is semi-solid and relatively softer. So whenever this head moves, it is separated from the skull. So because of the inertia of the movement, the brain is supposed to remain in position or cannot move with the skull if it moves suddenly. But this is prevented by this force of the dura mater, who separates the different parts of the brain and carries the brain with the movement of the head. The subarachnoid space is sponge-like. So this is working as the cushion effect around the brain and preventing the injury from the lumbar puncture is a procedure by which we can collect cerebrospinal fluid from the subarachnoid space. Already I have discussed the cisternal puncture. We can puncture the cistern magna and can get the CSF. Previously I have discussed, and that procedure is usually done in case of the children. But in adult we do the lumbar puncture. You see, after the lumbar 1, this spinal cord extends, and I have explained and shown that this may be the problem the existence of the spinal cord is a problem for collection of CSF. So this may be a bit beyond the lumbar 1, so lumbar 2 is also not safe. So it is better to collect the CSF placing the needle between the spines of the lumbar 3 and 4. So this is the area I will indicate by the arrow. This is the area you see. So needle will be in the subarachnoid space, and this area contains the roots who are hanging, and these are the cord-like. So the needle tip cannot injure them, easily separates them and can exist in the subarachnoid space, and easily we can collect the CSF to analyze that clinically. You can see the right lower figure, the needle tip is in the subarachnoid space, and if we are interested to inject some medicine into the subarachnoid space, we can easily inject that through this area. And this procedure, clinical procedure, is known as lumbar puncture. So lumbar puncture, we can collect the CSF, and usually done in the adult, and we can inject medicine like anesthetic medicine to give an anesthesia to a part of the body, not the general anesthesia. Local anesthesia can be given by injecting some anesthetic drugs here. Also, we can give other medicines through this way. Thank you very much for remaining with the presentation.